wanted to show you just posted the key for the exam. Notice that, so what I do whenever I have any class that I have open-ended questions, I scan all of the questions first before I have like my official grading key. Because I can expect something, I can have my own key, and then I don't know, students with full credits who give these points, and then if nobody has them, then it's clear I didn't address that. So it's basically kind of what can I get from everybody. So keep in mind that, well, math is math, right? So that's going to be just one single answer. But even if you don't have the correct answer at the end, that's why I wanted you to have some of the work there. So even if you miss, and because questions 1 and 2 are linked, if you have one incorrect, 2 might be incorrect. So that's why I want to see the work, because I don't want you to just get 0 points for 2 if you were like heading on the right direction, thinking properly. So all the answers are going to be here. Again, I tried to grab, like summarize almost everybody's, or kind of see if it's not exact, you know, again, getting everybody. So soil, roots, the atmosphere. So I just have, you were not required to have any of these factors affecting resistance, but some of you mentioned that to explain depending on like the types of soil and stuff like that. So I just put it there. Just give you some more information about it. And then, you know, the graphs. So some of you have it like this one, some of you have it like that. So I'm saying, kind of trying to address, you know, I will accept this as long as this is here. Again, if your graph doesn't reach any of these but has a similar trend, it's possible you will get some points. Again, everybody has different graphs, so I try to put all of that there. Similar to this one, I'm saying, like, you know, the important thing is that water absorption kind of couples with transpiration. And so, you know, you'll have time to read it. Of course, whenever I finish grading it, you will have comments in your exam. And then these are for the 4 optional questions that you had. So I also have. Okay. So hopefully, I think I will be able to finish grading by Friday. And then, you know, I'll give the exams back to you. And then every time, way worse is I give the exams back to you. The moment you have them back, you have a week to, you can talk about the exam with me anytime. But if it's about pre-grading, you have a week from the day I give them back to you. Okay? So you can check that. And so we're not going to talk about water anymore. So now we're talking about everything else in the plant. So I'm having some pictures today, and whenever it's time for the pictures, I'll take the lights lower. So we're going to talk about symptoms in plants, and that's just hard to see. But before we get there, it's not necessarily part of, you know, the reading. But have you ever heard of the Green Revolution? Does anybody know anything about it? So the Green Revolution. So, for example, what can you tell me? What do you see in this graph? Also, for the ones in the back, this is U.S. corn grain yields trend since 1866 to 2016.
Significantly higher.
Yield. What can you tell me from that?
Comparing yields increased significantly since the 1860s.
Yeah, so, you know, clearly 1860s, there's an increase. And another thing is, when do you see that? So this is kind of pretty flat, right? So clearly, something happened here. Because things were kind of on the same place, because his yield were just counting, cultivated area over whatever is being produced, this is normalized, but something happened here. And what happened here is what we know as the Green Revolution. So the Green Revolution started in the 1950s, like officially, and expanded until the end of the 1960s. And so you kind of see here, and then it continued increasing because, well, you're going to see that. But so the father of the Green Revolution is this man. His name is Norman Borlaug. If you ever Google him, and he actually was like a big person behind the International Wheat and Maize Developing Center in Mexico. And the idea of the Green Revolution is many things happened, but that was the time when scientists started thinking, let's not just be content with what we get in our backyard. Let's start making it better. We clearly need more food. That's when people started thinking, okay, we need more food. So how do we make it better? So they started breeding to develop high-yielding varieties. Also, the chemical fertilizers were not a thing that were used before. So maybe people were doing, crop rotation and maybe some organic matter, but not the plants that we currently have now for fertilizers. Also, irrigation was improved or maximized. So, it's not let's just wait for whatever rain brings us. Let's see how we can also. So put all the ducks in a row to make it better. And then after that, well, we have a combination of higher yielding varieties and genetic manipulation and all of that. So clearly just yields continue increasing since then. So actually he won the Nobel Peace Prize in 1970 because, so clearly the world recognizes this revolution as something that is before and after. Okay, so now that we've talked about the importance or more like how scientists started understanding, okay, what the plants need and how do we make it better, that's what made all of the possibilities of improving yield. So what can you tell me about this graph?
Fertilizers going up since the past.
Yeah, so we have higher yields, but also we've been using more and more and more fertilizers. That's how we can continue increasing yields. So it's not just genetic manipulation, it's also fertilizer, right? And the cost of fertilizers also is increasing a lot. Why do you think the cost of fertilizers increased as we use more fertilizer?
It's really hard to find cheap ways to fix nitrogen.
Yeah, so basically you use, the more you use something, the more you have to produce it. And as we're going to start discussing today, there's really no cheap way of producing fertilizers, really. And so basically just, I was kind of intrigued about what happened here. And we're going to talk more in detail when we talk about climate change. But I just found that there was a huge financial crisis in 2007, that apparently there were droughts and rising oil and fertilizer prices. Actually, that's when biofuels started being produced, and biofuels increased the prices of oil food. We're going to be talking about that. Clearly, you have to think more biofuel at the beginning, and if you don't do it right, it's worse. And also, the entire world started getting more food, and so the entire world started demanding more diverse types of food. Now we want to eat apples all year round. Now we want to eat oranges all year round. Now we want to eat everything all year round, and we want it to be pretty and fresh, and we don't want to spoil anything. So that's all driving prices out. So a couple of important facts that I want you to keep in mind is half of the energy that is used in agriculture is related to nitrogen. Production, distribution, and application of nitrogen fertilizers. Half of the energy that we use. So that's a big, clear. If we could find a way to make plants acquire nitrogen in more organic ways, that would be significant increase in the cost. And also, because you're going to see one of the most limiting factors that is also phosphorus. Phosphorus for fertilizers is obtained mostly from non-renewable sources like phosphate rocks. So clearly, you're digging, extracting something. It's not renewable. Yes, we can apply more, but where are we heading at some point? Has to be better. So more fertilizer is applied than what plants use and need. So if it's so expensive, why do people apply more fertilizer than what the plants need?
Is it like future soil health? Just like keeping more nitrogen in the soils?
So like,
So after the crops grow and like use what they need, there'd be like some left over. So like the next like planting would still have like some.
well, yeah, so in that sense, that's correct, but that is more applied after the plants are gone, right? So, yeah, but you also have to, that's why you also have to leave some debris and stuff there over wintering for to replenish the soil. But at the moment of growing the plants themselves, yeah.
It's also they're just trying to like decrease the competition between the plants for trees.
Okay, so, well, yeah, so that's one thing, right? Plants cannot move, so they are competing for nutrients as well, so you are applying more. But a couple of things that are important to keep in mind is remember when we talked about water and how you can, when it rains or you irrigate a plant, some of that water just gravity does its job and that's it. And there's so much water as soil can retain. Well, there are so many nutrients the soil can retain. Okay, that's one thing. And also, if you fertilize soil and then it rains, some of that is going to associate with the water and it just goes. So more fertilizer is applied well for many reasons, but a lot of it is going to be drained into groundwater. Many of those minerals are going to be just drained. And then many of them are going to remain associated with soil particles. So soil is very complex, has many particles there. So I cannot say, okay, if I apply 5 grams of nitrogen, there are going to be 5 grams of nitrogen available to the plant because depending on the composition and microorganisms and the soil and the pH and all of that, you might only have a fraction of that nitrogen available for the plant. Okay. So there's a lot of fertilizer that stays in the soil that again, if associated with soil particles, even when it's there, kind of what happened with the plant, even when it's there, it's not going to be accessible to the plant. And also, if you have a lot of drainage into groundwater, then you have eutrophication, and then the lakes have a lot of nitrogen and algal blooms and all of the ecological problems that are associated with excess of some of those compounds in water bodies. So when we talk about plants, what are essential elements?
It's elements that the plant needs that it can't produce itself, so it has to get from somewhere else like the soil.
Yeah, so elements that the plant cannot get from anywhere else, that it has to get from soil. And so basically, if one of those components, it's essential because if one of those components is missing, plant health is going to be affected. And basically one way I like to see it is if you give water, sun, and those components, the plant can thrive. Plant won't need anything else. So some components are important but are not essential, and some of them are not essential. So basically, the plant needs those building blocks to survive. If you give that and water and sun, plant is good. So this summarizes, this is table 5.1 in your book, and so summarizes, essential elements. So clearly the ones that are needed the most is hydrogen, carbon, and oxygen. Period. And then elements can be divided into macronutrients and micronutrients. What makes the difference between macronutrients and micronutrients?
Is it the difference between like which one the plant needs and excess, so kind of like the difference between essential elements and then like aren't they trace elements or something like that?
Yeah, so it is relative to how much the plant needs, but all of them are essential. Okay. So yeah, so all of these ones are essential, but you're correct in the sense it's relative to how much is not that the plant needs as in the soil, but if you examine plant tissue, how much of that relative to everything else you would find in that plant? Basically, how much of that element is present on an average plant tissue? Okay. So the most you're going to have is probably hydrogen and carbon because that's in every element we can think of. So, and then molybdenum just because they just use one that's very little, so they use, well, let's use it as a standard, so everything is relative. So basically, if the plant tissue assimilates a lot of it and it's in plant molecules, it's macronutrient. If it's in small amounts in plant molecules, then it's micronutrients. But this is kind of hard to grasp, so people don't really like to use this so much, again, because plants can vary and all of that. So that's not the best classification. We're gonna see kind of like an easier way to understand them. But one of the things I want you to get from this is that nitrogen, potassium, and phosphorus are the main, so next time you go to Lowe's, go and find any fertilizer. Most likely, this is what you're going to find on a fertilizer. So we're gonna talk more about, nitrogen and phosphorus later in the semester. But whenever you grab a fertilizer, you're going to see most of them will have 3 numbers, and they are not telling you this is this. They assume you know what it is, right? So it will have 3 numbers, and they stand for N, P, and K. So it's the proportion, relative. So most fertilizers are going to contain nitrogen, phosphorus, and potassium because those are the elements that most soils are deficient. So the way I also prefer classifying plant nutrients is because just putting into perspective what they do for the plant, and then that kind of tells you groups and importance of these elements. So what can you tell me about group 1?
That's using a lot of protein synthesis.
Yeah, so group 1, nitrogen and sulfur, right? So part of carbon compounds. So, of course, carbon compounds can be many things, but when you think about amino acids, protein, coenzymes, so enzymes and amino acids, basically you need, or you can think of proteins is going to contain nitrogen and sulfur. Of course, we're not counting carbon because remember the carbon is going to come from CO2, so that you don't have to feed carbon in a fertilizer. What can you tell me about group 2?
They're important in like energy storage and structural integrity of the plant.
Yeah, so energy storage, structural integrity. Again, one thing is keep in mind is like energy storage, you kind of need these compounds to store energy, so it's like a kind of, again, arbitrary classification. But so structural integrity, so when you think phosphorus, you think phosphate, you think DNA, lipids, ATP clearly. Also in cell wall, so boron is a very important component in meristems and cell wall integrity, so just overall integrity and energy. What do you see in common for group 3?
Maybe aren't most of these used for like transportation and like across membranes and reactions in that sense?
Yeah, so, ions, they are all ions, and so, they are, so cofactors, right? Many enzymes need cofactors to work properly, so some of them are second messengers, so calcium is a very important second messenger. Maintenance of cell turgor, so potassium is important for that. Photosynthesis, so whenever we talk about photosynthesis, you're gonna see magnesium is like the central ion in a chlorophyll ring, is required for chlorophyll production, so essential for that. And then in group 4, what do you see in group 4?
Is it energy transformation?
Yeah, so basically redox reactions and electron transfers. So they are part of protein complexes. So iron is very important as well. We're gonna see in photosynthesis and molybdenum. All of them are just important also in nitrogen fixation and mobilization. So more like redox reactions. Then again, it's hard to just isolate them from there. But I guess for me, it's like a glory kind of group, like, okay, let's put everybody else in here. So, okay, so we have classified them. But so let's think that you are, I don't know when they figured this out, but let's say 60, 100 years ago, and you don't know what essential elements are. So, well, it's clearly not 100 years ago because it was like sort of around the Green Revolution. So you want to make sure your plants in your garden have everything they need. And you're kind of being you're not wasting any components, so you want to give the plant what the plant needs, but you also don't want to waste money on things that are not essential for the plant. So you're trying to figure out what is essential for this plant to grow. How can you determine if I'm ever plant to grow?
Just do what we're doing in lab right now, basically, where you take the element out of the nutrient, and feed them, and see what happens.
Yeah, so basically you feed the plant everything else but one nutrient, right? You cannot take 2 nutrients away or 3 because then, of course, from experimental design, you have to have all variables controlled and you're only testing one, otherwise you wouldn't know the effect that you have. Another important thing, why do you have to do it? Like, why did scientists have to do hydroponic culture to do this?
Because you can't control which elements are naturally in the soil.
Yes, because finding the soil composition is very difficult, and again, because then you don't know which elements are going to be associated with soil particles. So again, you can be putting 5 drops of this in the soil, but are they available to the plant or not? So maybe you put it there, but you have no way of figuring out if the plant is actually using them. So it's the effect that you're seeing because the element is not essential or just because the plant couldn't get it. So that's what started hydroponic cultures. And so one of the things is whenever you do hydroponics, and of course very basic, is you always have to be adjusting nutrients. Because clearly the plants are going to absorb them, so you have to maintain the solutions. Constantly adjusting pH and providing oxygen to the plants. Okay, so what can you tell me about the basic hydroponic growth system? Just the basic one.
You just put like all the essential nutrients into like the solution, and then like have the plant just like drawing all the nutrients straight from the water.
Yeah, so you have the nutrient solution, the roots are there, clearly no soil or solid substrate. And then you have to supply aeration, so air bubbles. So again, there's really no movement in that solution. The plants there's respiration going in the roots, so you want air to be available because respiration affects pH, and also because you need oxygen for the plants to continue doing aerobic respiration for that. How is that different from the neutral growth system?
Instead of having it in a bath, you just have like the solution running over with the roots. It affects like aeration. You don't have to be like you still have that like an air pump in the system, but you don't need like air like bubbling through the roots.
Yeah, so what was it that you said about the bubbling?
It was just like you don't need air bubbling like through the roots. So you still have an air pump, but it's like not as the aeration isn't directly through the root.
Yeah, so what you see is the roots are not submerged like here. There's just this thin film that flows, and then gravity gets for it. So you're actually putting air to put air into the solution, oxygen into the solution. But then the roots themselves are submerged, so there is some good aeration in here. An important thing in this case is that, yeah, so basically it's more complex because, there's gravity here doing the work, but you also have, just aeration and how much aeration is present is very important in this first one, because you don't want to stress too much the roots. In this case, the roots are isolated from all of that bubbling, so relatively, this is more complex. This is more complex, but this makes it easier. What about the ebb and flow system? I think the name is very self-explanatory, but what is the ebb and flow system?
You'll drain the water from the top to aerate the plants, and then you'll eventually put water back in so the plants are still getting nutrients.
Yeah, so you put the nutrient solution in here, let the roots sit there, and then drain everything, and then everything floated, and then nothing present. One of the kind of things about this system is that your nutrient solution has to have much higher concentration of nutrients because the roots are constantly exposed to the nutrients. So whenever they are exposed, they have to be able to uptake the nutrients. So nutrients have to be at a higher concentration just because they aren't always exposed to these solutions. And then this last one. How is that different?
Instead of the roots being like submerged and then like not submerged, they're just constantly sprayed with like the nutrient solution.
Yeah, so instead of just not submerged, you have this constant spray. And also an important thing is clearly there's air happening there, right? It will depend on the type of plant that you have, the type of environment that you have. And so in this one it is even more important that the nutrients are at a higher concentration than the plant needs. Because again, you're just spraying. So that's also an important aspect. So The solution, or more like kind of universal solution, is known as Copeland solution. And it's, to say the least, it's horrible to prepare, right? If you think about it, you have to make all of these stock solutions and then mix them in the right ways, and you have to be adjusting them. You can buy it. It's very expensive to purchase it. So if you are running like a quick experiment, you might be able to get a few grams for $50. But if this is like what you do for living, then you're just gonna have to pay so much for that. Okay, so let's talk about the Copeland solution. There are two different sources of nitrogen here. Why is that? Why is that important for plants?
So one of them is a cation and the other one is an anion. And the mixture of both of them helps keep the pH from rising too rapidly.
Yeah, so it's very important to have them. So plants, if you pause for plants would do the ammonium over everything else. Fungi and plants, make them choose ammonium is the way to go. That's their favorite source of nitrogen. But they also need different charges to maintain the pH. So what happens, and we're going to talk about this extensively, we're going to have like 2 entire classes about nitrogen things, so I'm not going to go into details. But basically what happens is when the plant uptakes ammonium, tends to release hydrogen ions. And when it uptakes nitrate, then releases hydroxyl ions. So clearly if you have one or the other, like in the case if you only gave it this type of ammonium, the favorite source of nitrogen, you would decrease pH, and then if pH in the soil or in the cell becomes too low, well, you know what happens. So there has to be a balance. Also, whenever we talk about transport of solutes, have you ever heard of symport and antiport transport? So those electrochemical gradients are essential for driving nutrients in and out of plant cells, and they rely on gradients, sometimes pH gradients. If you perturb that gradient, it's not just nitrogen, but almost everything else is going to stay either in or out because they rely on that gradient to move other things. Do you know what EDTA is? And, well, let's start. And or why would that be associated with iron?
I could be completely wrong with this, but I'm pretty sure that EPTA, you have to have the iron, keep it so that it's not constantly reacting, because it wants to, I think, oxidize almost immediately. So I think you have to add that to stabilize it, or otherwise it'll react.
Yeah, so it's for preventing iron from interacting with other things. How do you call those general class of compounds that kind of associate temporarily by non-covalent bonds with certain ions? And instead of like keeping them in solution, very specific word for that. I think it's LO3 or
Chelators?
yeah. So I think you say chelators, but I could be
No, I'm
wrong.
pretty sure I pronounced it wrong.
Oh, me too. Believe me. So I'm gonna write it here. And then So chelators and this EDTA, you're gonna see it a lot, like whenever you're in the lab and you're, for example, whenever you set up a PCR, the buffer that you include in the PCR usually has EDTA because the cofactor for Taq polymerase is magnesium. And so it prevents magnesium or other ions from associating with things that are not what they should be associated with. So iron is one of these things that can very easily just go along with something else instead of remaining available to the plant. And it's very, very susceptible to pH changes. Any tiny pH change, and it will precipitate out of solution, becoming unavailable, or will associate with other things. So, kind of addressed this already. So these chelators, this is not really EDTA, but it's a different chelator. And so basically what you're seeing is that our molecules, very complex molecules, and here they are kind of protecting that iron, associated temporarily with that iron by non-covalent bonds. So it kind of protects it, keeps it in the soil. And then sometimes what happens is the plant can uptake this whole thing, right, in this case. Or basically, so it's less clear, the book says that it's less clear what happens once the plant uptakes that, but it seems it dissociates and then the iron is kind of handed into other components where the plant can deal with it in an easier way rather than competing with soil particles and pH changes. So that's what they do. And iron is one of the limiting factors because of pH. So changes in pH in the soil are very important. So, which factors determine the deficiency symptoms for a particular element? So, let me put this in a different way. You have deficiency for a particular element in your own body. What determines the kind of symptoms that you show? How is that related to the element that you have a deficiency?
If you have a compound that requires an element to produce, like say an amino acid, they can't produce that amino acid. So whatever that job is that amino acid would do is no longer being done. So that would be replicated in something.
Yeah, so the function of that element or the job of that element in cellular metabolism, so in this case in the plant. So what that particular element does in the plant. Now let's talk specifically about plants. What other key factor determines the symptoms that you see of nutrient deficiency?
Are you just talking about how like the plant looks? Like you can just kind of determine what's wrong with it based off of what it looks like?
Yeah, so that's how the plant looks like, and that's a very important thing. So we're gonna see some of the most common symptoms in a minute, but so one of the things is whenever you wanna figure out what's wrong with a plant, and you're gonna see it once the lab happens. If you bring someone a leaf and you say, my plant is doing this, you cannot say anything. You actually have to look at the entire plant, and if possible, more than one plant. So, like if you go to like a plant diagnosis lab, sometimes farmers bring like a whole truck with plants, and it's like, well, that's more than what I need, but I'd rather have more than just the leaf. So you need to look at the overall plant. Why do you need to look at the overall plant? Why do you need to see more than one leaf to figure out the deficiency?
We talked about this the other day, but if it's like a whole leaf, that's where you're going to see the deficiencies compared to the leaves, because I can't remember the recent date, but I think it's pretty recent.
Yeah. So what determines if an element, if the deficiency starts in new leaves or in old leaves?
Is it like mobility of the specific element?
So mobility of the specific element in the form. Okay. So two things determine the symptoms. The role of the element, right? So what the function of that element in the plant, and the mobility. Can it be redistributed in the form or not? And so that will tell you, so whenever you're looking at a symptom, it's not just what I see, but where I see it. Relative to, is it new growth or is it old growth? Okay. So I think this is where, yes. So if you cannot, if a plant cannot mobilize it, element X cannot be mobilized in the plant. Where would you see the symptoms? Yes.
In young leaves?
Where?
In younger leaves?
What?
Older leaves, perhaps already had the element from like when that was not a deficient element. So when you form a new one, and you're deficient, you can't move that to the new leaf.
Yes. So one of the things is that, so when we think of, photosynthesis, so let's take it, like a family. When a family has, babies and grown-ups, the grown-ups go to work. Eventually, the babies will be able to go and bring money, but not yet. So when younger tissue is developing, they are more uptaking nutrients from the older leaves. They are not fully developed, so the rate of photosynthesis in younger tissue is limited. They cannot fully, they are actually more called sinks than sources. They are more uptaking elements from the rest of the plant until they are fully developed, right? So for a while, even when a leaf is young and green, it might have some photosynthetic capability, but it still is a sink and relies on food from the grown-up leaves. Okay? So also the grown-up leaves, depending on their stage, will eventually senesce. So again, like the example of the family, if a plant has developing leaves and has the grown-up leaves, and it has deficiency, you think about a family and there's shortage of food, what do the parents do? They prioritize feeding the kids over feeding themselves, right? So that's what plants do. If there's lack of nutrients, if there's deficiency of nutrients, the plant will prioritize feeding the little ones, because that is the future of the plant. They are almost heading out. So the plant will try to mobilize those elements into continue growing the younger leaves and moving it into the younger leaves. So if the element can be moved and there's a deficiency, if it can be moved, then you will see the deficiency first in the older leaves because they are prioritizing the little ones. However, if the element cannot be moved through the phloem, then the big leaves have no choice. Even when they want to feed the little ones, it cannot be transported through the phloem. Now, this is one of the parts in which we don't know yet what makes an element mobile or not. So this is one of those cases in which the only thing you can do is memorize. And every time I teach this class, every year I have to memorize it again. So I'm not saying there's an easy way. And I don't have tips for you to memorize it. So telling you, we're not going to talk about all of them. So there's going to be, this is in your book, so I wouldn't worry too much about taking these notes. Just make sure, there's no way around memorizing. You might have better ways. So what I decided to do is group them in pairs where the symptoms are very similar and the major difference is mobile or immobile. Okay. Now, an important thing. The symptoms we're going to be looking at are very simplistic one way. So, for example, symptoms of nitrogen deficiency can be so many that I'm going to give you like the Kirsch's typical nitrogen deficiency symptom. Okay, so what we're gonna see is not the only thing that, like, when we talk about sulfur deficiency, clearly it's part of protein, so it's not going to be a single type of symptom. So we're gonna focus on, like, clear textbook picture of nitrogen deficiency, sulfur deficiency level. Another important thing is we're not really going to talk about that so much, but I'll show you the picture at the end. Deficiency of something like in our bodies, even when you have deficiency of, let's say, vitamin C, that is affecting other things, right? So deficiency of one element can affect the availability of other elements or uptake of other elements. For example, if you don't have enough nitrogen, you're not going to have enough proteins, and probably uptake of every other element is going to be affected at the end, right? So trying to keep it simple is much more complex than that, but we're going to keep it, again, textbook picture of each one of these deficiencies. So we kind of already explained that. So, mobile elements, other leaves, because again, if there is deficiency, then the other leaves are going to try to give food to the young ones. So we're going to talk, and we're going to talk about 1, 2, 3, 4, well, not 0, 1, 2, 3, 4, 5, 6, 7. Or actually 7. We're going to talk about these deficiencies. Okay? And you're going to have the slides. So what I did again, textbook picture, is group them so that you only have to memorize or recognize 4 or 5 symptoms, typical symptoms, and then some of them, the main difference is if the element is mobile, you're going to see those symptoms first in mobile, in other leaves, or in mobile here. So let's start with nitrogen and sulfur. The symptoms for nitrogen and sulfur deficiency are basically overall pale leaves. Okay, so you can see here, light and green, so nitrogen, well, one is mobile, one is immobile, we're going to see that, so hopefully so nitrogen is mobile, sulfur is not. So overall, pale leaves. So if a leaf, the way I can summarize it easily is, if a leaf should be green and is kind of yellowish, pale green, the overall leaf, think of it nitrogen or sulfur deficiency. Okay? And the key is going to be where you see it. Because one can be mobilized, the other one cannot. So nitrogen deficiency, this would be a progression of nitrogen deficiency. So here you're comparing, this is going to be a normal leaf, and overall there's really like no pattern. The whole entire leaf starts becoming yellow. That's known as sclerosis. So it's becoming yellow, or pale green actually, overall. Here you can see, well, sort of, can see a nitrogen deficiency here. This is in corn, and so you can hopefully see where you see in the plant is going to tell you if it's nitrogen or sulfur deficiency. So in this case, and this is how we're going to try to show, so before we next class are going to have an activity where you are going to identify it, and we have better pictures than this. But sulfur is immobile, right? So in this case, you can hopefully see that if you compare this, this would be lighter. So the young leaves start being pale versus the old leaves. So that tells you this is sulfur. Because nitrogen is mobile, then the older leaves are going to be pale instead of the young leaves. That's how you can tell. The next group is going to be iron and magnesium. So what are the key things about iron and magnesium? You see it's pale leaves, but the veins remain green. So the overall leaf is kind of yellowish, but the veins are remaining green. And so iron And, if you see iron deficiency in young leaves, what does that tell you about iron? Can you mobilize it or not? So it's immobile, right? If you cannot bring it to the young leaves, then it's immobile. Magnesium is the opposite. You see it in all leaves, that means it's mobile. Also, I can tell you, well, I'll finish this, but this, we're gonna do an exercise in this next class, and this is 25% in midterm. It's gonna be pictures, and you identify the deficiency and where it's mobile or not. Yes.
I guess I said the iron is.
Yeah, so I have another slide here. So now hopefully you can see the difference. So it's pale, but the veins remain dark green. Also for the exercise that we have next class and for the exam, I will make sure I stick to the main symptoms that we talked about previously. Okay. So magnesium can be blue, iron cannot. So I put it, so now you know younger or older leaves. I just wanted to show you, like if you see pale green veins, it's gonna be either magnesium or iron, and then depending on where you find it in the plant, or not. Okay. Potassium. So potassium is doesn't have like a counterpart, but the yellowing starts in the borders of the leaves. So not so much overall, it's mostly the tips. And so potassium, I wanted you to compare it to nitrogen because we saw nitrogen a few minutes ago. So remember how nitrogen was the overall leaf, all of it becoming pale? Potassium is the tips, is what starts becoming yellow, and then it progresses down. And it's harder to see, so of course, again, in the example and in the exam, I'll try to show you, like clearly the tips are yellow versus the rest of the leaves.
With old leaves.
If you can move it, it will be old. They can feed the little ones after old. Phosphate, old leaves, so old leaves tells you small. And there are many different, actually phosphate is very complex and it overlaps with many things. So I took the only thing that doesn't overlap with everything else. That is, the leaves can turn purple. Sometimes nitrogen can do that as well, but so in this class we use purple phosphate. Okay. And it was hard to find purple leaves, but I wanted to give you the idea of, again, can be for other reasons, but in this class if you see purple, we're gonna talk about phosphorus deficiency. Okay. And the last one is calcium. New leaves, which means young leaves, which means that you can mobilize it, are misshaped or stunted and no stunted. And so kind of misshaped. So you see misshaped leaves, and this is younger tissue, right? I mean, so this is kind of an idea of how I'm going to show you if it's younger or older. This is younger tissue, this is older tissue. So younger tissue, and it doesn't have counterpart for the symptoms we're talking about, but it's going to be kind of misshaped. I mean, I have to say it's hard to see, especially because that window is there. But whenever you get home, if you can see these better in your screens, you can hopefully see these lower ones are older leaves, and these one the top leaves are young, are the leaves with the deficiency. So in that case, because there's overall fluorosis, you remove this is nitrogen and then. Okay, so make sure you review this for next class. And next class, this is what we're going to be doing. It's a worksheet, all the lights off. And it's going to be a simulation of the exam. So you're going to have about a minute and a half, I'll put a picture, and you have to identify. You can use clearly your notes next class, not the exam, but it's going to give you, that's how we can do it. Okay, then a minute for you to figure out what kind of element, model and model, blah, blah, blah. And so that's what we practice for. Okay, so make sure you review that. Let me know if you have any questions, and I will see you. What is the